So it's now my distinct pleasure to introduce the 2022 Gold Medal Award winner. And just a few words about the WMIC Gold Medal, which is the highest honor within the society. It is awarded for a major research contribution to the field of molecular imaging within the scope of our society's purpose. And it is just truly an honor, and I think even overdue, that our 2022 Gold Medal Award winner is Anna Wu. Anna is the professor and chair of immunology and theranostics at City of Hope, the co-director for the Center of Theranostic Studies, and the co-founder and chief scientific officer at ImagineAb. And I remember when this company was, I don't know how many years ago, was a few employees and is now like a major player working with big pharma. Anna was also the president of the World Molecular Imaging Society from 2013 to 2014, and she's also a fellow of our society. She's won many awards. These are the ones I think she's very proud of. The Suffrage Science Award in 2018. She's a fellow in the Society of Nuclear Medicine and Molecular Imaging. And this year, she won the Women Leadership Award and gave such a wonderful talk on what that meant to her yesterday. It was wonderful. Anna's research focuses on engineered antibodies for targeted delivery of radionuclides for imaging and therapy, and she's published over 200 papers, chapters, and reviews in molecular imaging and therapy, and she has 12 patents, and she's also a grandmother, probably many times over now. So with that, I want to welcome Anna to the stage.
Thank you so much, Carolyn. And thank you, everybody, the Society, the Awards Committee, all of you. There is no greater honor than to be recognized by your peers. And to me, it's just amazing to be standing up here in front of you and giving this talk. And I also wanted to say what an amazing few days we've had. I think you all saved up your science over the past 3 years, and it's just all come out, like a storm surge. So I'd like to talk to you today about, and I'm going to steal a page from both Marty Pomper's talk earlier this week and also Joe Ackerman's use of the Wayback Machine, because I want to talk about how we put together imaging and immunology. And in order to do this, I have to go way back. And I thought you might be interested in seeing, what were the roots and how did we get here in terms of being able to use immunology to image, and then to image immunology. So first of all, these are my disclosures, and the relevant one, of course, is that I'm a board member, shareholder, and consultant to ImagineAb. Okay, and this is what I'd like to talk about today. I've used this phrase before, putting together antibodies and antimatter to give you immunoPET. And so what I'd like to talk about today are just walk you through why I decided to work on antibodies, why we should all be working on antibodies, why radioisotopes, radionuclides, how to put them together, and then finally talk about how we can use what I call applied immunology, which is immunoPET, and apply it to immunology. So why antibodies? I think I've always been drawn, and really since I was in college, the unparalleled diversity of antibody binding. It's thought that the naive antibody repertoire is estimated to be up to 10 to the 12th. That's how many B cells you have in your body. Each one can have a distinct antigen recognition capability. And I remember when I was in college, this was the great mystery, because there wasn't enough DNA in a cell to code for that many different antibody genes. And it really took it was really actually just groundbreaking when Susumu Tonegawa figured out that the DNA actually gets rearranged to give you all this diversity of antibodies. And for that, he, of course, won the Nobel Prize. But at the time, it was heresy. It was absolute heresy, and so exciting. The other thing, next thing about antibodies, is that they really are a platform. So if you look at the antibody molecule, whoops, wrong button. There we go. only a small fraction of that large antibody protein is actually involved in specificity and target recognition. And those are the complementary determining regions shown here at the tip of each arm of the antibody. But what that means is the rest of the antibody, the constant regions, the framework regions, are fairly constant. So the properties of overall antibodies are generally predictable. So you can really, you can vary the specificity, but in terms of how you make them and how you use them, it's a common platform. You don't have to reinvent the wheel each time. They're also very malleable, and this is where I come in. I came in as a protein engineer, and through protein engineering, we have extensive control over the properties of these antibodies, and I'll go through some of the things that we as a field are able to do to make these antibodies more useful for what we want to do. Translatable, very important. And I think what's happened over the course of my career has been that therapeutic antibodies have really become completely mainstream if you look in, especially in oncology, but other fields as well. So there's this tremendous infrastructure and knowledge how to make antibodies for clinical use, how to scale them up, how to deliver them as highly effective products. So all that infrastructure for manufacturing, clinical trials, FDA approval. FDA knows what to do with antibodies, so that's all in place. The next bullet really is, it could be a feature or a bug, and that is the long circulating half-life of intact native antibodies. It can be a feature in many therapeutic settings because it's nice to have long circulating half-life. You only have to administer your drug once every 3 weeks, or with some of the half-life extended versions, once every 3 months. So that can be considered a feature. But for those of us in the radioactive world, it's actually could be a bug because you've also got that radioactivity circulating for equally long time. So we'll talk about that. And then finally, I guess this is a bug, but it's not a deal-breaking bug, which is that antibody specificity in vivo is going to be limited to cell surface targets. Once antibodies get inside cells, they quickly turn into food. So that's something to keep in mind, but there's plenty of interesting targets on the cell surface because that's where the cell interacts with its environment. There are receptors, there are transporters, there are adhesion molecules, all kinds of conversations going on at the cell surface. And so if we can figure out which are the key molecules that are telling us what those cells are talking about, those could be very interesting targets. And we also know, though, that antibodies are very large compared to something like fluorodeoxyglucose or some of these small molecules we work with. That can be a bug because it does mean that they can be limited in their ability to penetrate tissues, and particularly tumor tissues, which have very poor perfusion, circulation, et cetera. So those are things to keep in mind, but we can work on all of these. So the way we work on it is through antibody engineering, and I just want to walk you through the process because most of us in the field started with mouse monoclonal antibodies, and if we want to use them in patients, they're foreign proteins, they're immunogenic. So this is just a quick snapshot of a mouse antibody, fully mouse antibody here in pink, and then we can make chimeric antibodies by swapping out the variable regions of these antibodies. We keep the mouse binding sites in pink, but change the constant regions to human. We can also go farther and just swap out those CDR regions, those loops that make the binding site, and make a humanized antibody, or go all the way and make a fully human antibody. So this is something that we've done in our lab for years. And so, for example, here's a list of some of the antibodies where we've started with a mouse antibody against CEA or PSCA or NCADHERE and humanized it so that we can translate it. We can go even further and go to fully human antibodies, and one of the ways this can be done is through phage display libraries. So instead of libraries of B cells with different antibodies on them, we have libraries of bacteriophage with different antibody binding sites on them, and can select them in the laboratory and quickly go straight to a human antibody. So again, this has been a very powerful technique in the field and something that we've also used to generate novel antibodies that could be directly translated to patients because we have a library of human antibodies that we start with. Okay. And then, of course, my favorite theme, which is production and the use of engineered antibody fragments. And by taking the intact antibody and just rearranging or reordering or keeping the activities that we want, the functions that we want, and getting rid of the ones that we don't want, we can really tailor these molecules to our final application. So for example, through protein engineering, we can have control over the valence of our molecules. Native antibodies start out as bivalent, but we can monovalent or multivalent or multispecific. We can control the molecular weight of these proteins. We can steer the clearance by taking advantage of that 60 kilodalton approximately threshold for first pass renal clearance. So as most of you know, smaller things clear quickly through the kidney. Larger things like antibodies will clear more slowly and end up in the liver. We also have these FC interactions. There are receptors that interact with those constant portions of the antibody, either for biological effect, the receptors, the FC receptors on immune cells, or also having an impact on the circulating half-life of these antibodies through interactions with FcRn receptors. So we can control those or edit them out in our case. So that way we control the pharmacokinetics of these proteins, so we can start to get things that clear more quickly than native antibodies. And this is actually shown here in this slide, just showing you our building block, which is a single chain variable fragment where the two variable regions are linked by a protein linker. We can make dimers called diabodies. We can make slightly larger molecules. I'll talk about the mini-body more. It's one of my favorites. By adding one of the constant region domains back, or we can have single chain FVs that have a full FC region, and we've heard about that a bit at this meeting, or intact antibodies. And importantly, we can end up with a family of molecules that have different pharmacokinetic properties. They do, and of course, a longer half-life in the blood does mean you will get higher absolute levels of tumor uptake, but you can also see that a quicker clearance rate. We can still achieve high uptake in targets of interest, particularly because we maintain the bivalent and high affinity binding, but coupled with much faster clearance, we have proteins that give us contrast at earlier times. So let's go through this a little more detail. Let's see. Oh, no, I have to change gears first before we put it all together. So, why radioisotopes? Long ago, even before college when I got hooked on antibodies, I actually got to work in a lab when I was in high school. I got a fellowship to work at the New York State Department of Health with Dr. Leo J. Grady, who was working on bacterial DNA, and he was working with Bacillus subtilis, and it had a latent bacteriophage called PBSX, and this was really in the dark ages, before DNA sequencing, if you can imagine that. And he wanted to study the relationship between the bacterial DNA and the phage DNAs, so what we did was we radiolabeled them with either tritium or C14. And this was really cool because we couldn't see this stuff, but we could follow it, and we could find it. It was permanently tagged in a way that we could find it. So that was my early introduction to radioisotopes. And that's why I like them, because you can find radioisotopes. You can count them. If they give off photons or high energy, you can image them. You can quantify. You can conduct quantification, particularly with one of my favorite modalities, which is PET imaging. Another reason we like radioisotopes or radionuclides is that they come in different flavors. And so we've heard about that throughout the meeting and also in Jason's highlights. Alpha emitters, beta emitters, gamma or positron emitters. So a variety of different emissions out of these radionuclides. And of course, with a range of physical half-lives and a range of emission energies and penetration properties, there's the periodic table. And you can see just how many goodies that Mother Nature has given us, but also importantly, our friends who have cyclotrons and nuclear reactors can give us. And another reason I like radioisotopes is that they go away. They're not around forever, and I will maybe comment a little bit on that later on. They're not with you forever. And we also have in parallel, there's been a huge amount of research in radiation biology, much of it focused on using, say, for example, external beam therapy, but also in terms of radionuclide effects on cells and tissues. So there is a very defined body of information and growing body of information about radiation biology and interactions of radiation with cells and tissues. And of course, I like radionuclides, radioisotopes, because they are translatable, because we can detect these ionizing radiation, we can detect it outside the body. unlike the challenges we face with some of the, for example, optical imaging approaches. So that's why I like radionuclides, radioisotopes. And I actually, again, want to go way back because I want to just a little bit of historical work. When I was first at City of Hope, I joined a group that was already working on antibodies that recognized carcinoembryonic antigen, or CEA, and actually Michael Neumeier had previously made a chimeric version of the antibodies shown here, chimeric T8466, with the goal of being able to translate this into patients. And that's what the group did. And so this is really, I think, an early example of the theranostics that we were doing back then. We would first radiolabel the antibody with indium-111 for SPECT imaging, and then change the radionuclide to yttrium-90, a hard beta emitter, so that we could do targeted radioimmunotherapy. So here's the group back in the 1990s. And you know, this is, we feel vindicated because this has now become something that's, you know, much more feasible and now being shown to be more, you know, truly effective in the clinic. Theranostic approach, to be able to use the same molecular targeting agent labeled with an isotope first for imaging purposes and subsequently for therapeutic purposes. Okay, and so what about the engineered fragments? Actually, I came up with the idea of this mini-body for therapy. So here are the design considerations. I wanted to make a single chain construct for ease of expression instead of having to co-express, and this was in the early days when it, you know, was a challenge, co-express heavy chains and light chains and have them come out in the right ratios and have all four of these chains assemble correctly to give you an engineered antibody. We just went for a single chain fragment. So you only have to make one piece of DNA, one piece of protein. It folds up, it self-assembles into a dimer as shown here. So it has two single chain FV fragments for high avidity antigen binding. We added the CH3 domain, both to add mass. We wanted to bring it above the threshold for first pass renal clearance, and also as a dimerization domain. Those CH3s come together with 10 to the 13th affinity. So you would just produce this one chain and it would just spontaneously form dimers. We also threw in a hinge, so we would have a covalent version of this for added stability. And the fact that we don't have the full FC region means that this protein is biologically inert. It does not interact with immune effector cells. It does not interact with FcRn receptors. It's just a vehicle to carry those radioisotopes. And we also were hoping that the fact it has a smaller size compared to an intact antibody, that it would show improved tumor and tissue penetration. So those were the design considerations for the mini-body. And this is an early image out of that original paper. It's actually SPECT images. It's I-123 labeled anti-CEA mini-bodies. And let's see, we have a mouse here with a small, large, medium, or small xenograft, CA-producing xenograft, imaged at time 0 or 4 hours or 19 hours after injecting. We had to sneak into the nuclear medicine clinic after hours with these mice and use the clinical scanner. So yes, we've come a long way since then if you took a walk through the exhibit hall. But we nonetheless got these images that showed us that yes, this engineered antibody fragment does target to CEA-positive tumors, and it does so quickly with rapid blood clearance. So, but I'll also point out that, you know, these are the properties you want for both therapy and imaging. For radionuclide therapy, which was our original goal, as you can see from the fact that included in that paper was projected tumor to blood AUC ratios for iodine-131 and yttrium-90, which are therapeutic radionuclides emitting hard betas. And we were trying to make a fragment that would show rapid tumor uptake and retention and overall optimize the radiation dose to bone marrow, kidney, liver, and other normal organs. But because we were, even though we didn't know we were a theranostics group, because what we did at City of Hope was to image first. Here are some early imaging studies, clinical imaging studies, showing that, you know, those same properties are ideal, potentially ideal for in vivo or clinical imaging agents. Because again, for imaging purposes, we want high tumor activity and rapid blood clearance, and we want to get high contrast at early times. So here are some clinical images where we either labeled the anti-CEA minibody with iodine-123, or indium-111 and imaged patients with colorectal cancer, and were able to demonstrate targeting and imaging of this fragment by SPECT, by planar imaging and by SPECT. Okay, what about PET? We all know the wonderful advantages of PET imaging. It's very sensitive, it's higher resolution than SPECT, it's quantitative, but when we entered this field, we also realized that we really had some challenges in trying to put this together, and this was, again, a long time ago when we did not have access to the radionuclides that we have today. So when you thought about putting together antibodies and PET, 99% of the work in the world in positron emission tomography was being done with fluorine-18, which, as you know, has a 2-hour physical half-life. So that became the irresistible force, and the antibody, of course, is the immovable object. It takes so long to get to its target and clear from the bloodstream, typically 1 to 3 week biological half-lives. How are you going to put these together? By the time the antibody gets where it's going, the radioactivity is long gone. But now I've shown you I have potential solutions on both sides. So one thing that's happened is on the supply side, the radionuclide side, we now have access to a much broader spectrum of positron emitters with longer physical half-lives, such as copper-64, zirconium-89, iodine-124, with half-lives from half a day to several days. And then I've just shown you that we have ways to engineer antibodies into these defined fragments that have shorter biological half-lives, such as minibodies and diabodies. So this is what I did my sabbatical year at UCLA. I started my career at City of Hope, as you saw, and was part of their incipient theranostics team. But Mike Phelps invited me to spend a year at UCLA, and he invited me to work with this brilliant young assistant professor who had just joined as an assistant professor, nuclear medicine expert, and that was, of course, Sam Gambier. But it wasn't just Sam, it was a whole team of people that enabled us to take the anti-CEA minibody, to radiolabel it with copper-64, which came from Mike Welch, Deb McCarthy, and their colleagues at WashU, and to conduct this kind of study, where we imaged mice that had either CEA positive or CEA negative xenografts in the microPET. At the time I was over there, this was the one and only microPET on the planet. It was developed and built by Simon Cherry, Arian Hatchuano, and their team of students and postdocs. And you could see that we were able to show by PET imaging now, and this is before PET-CT too, as you can see, that very nice uptake of the anti-CEA minibody labeled with copper-64 to the tumor, liver clearance, and as we had hoped and expected, this looked like it had great potential as a platform for immunoPET. And one other thing that we did early on, especially in thinking about these smaller engineered fragments, early on the concern was, with a protein, we're typically, initially we would be conjugating and radiolabeling, for example, through amine chemistry to random lysine residues on the surface of the protein. And you've got a very large antibody, you can get away with it because there's all those constant regions where you can tack on a chelating group and a radiolabel, and you won't impact the ability of the antibody to hit its target. But when we're working with these smaller fragments, such as these diabodies, we were concerned that we might accidentally hit key residues in the binding site or over-modify that protein and end up impacting its ability to bind to its target. So as protein engineers, we said, okay, let's engineer in something that we could do site-specific chemistry on. And so one of the things that I did my sabbatical year was to make these modified diabodies introducing cysteine residues either into the linker in the diabody or at the C-terminus. And you can see what happens when you put them on the C-terminus. You actually get a covalent cis diabody that you can reduce into these monomers and use those thiol groups for site-specific radiolabeling. And on the right shows, again, early microPET images showing cis diabody targeting to the CEA-positive tumor, again, in early and in late time points, showing that we're able to modify these proteins in a site-specific and stoichiometric manner and preserve overall antigen recognition and targeting abilities. So that was an important trend or important factor to have. And of course, we've heard a lot today about other ways you can do site-specific and very interesting but very defined modifications to proteins. So that really has been an important field. Okay, so now we have a platform, and this just summarizes a lot of work by a lot of people, same day and next day immunoPET, using either diabodies or I'm sorry, I keep hitting the wrong buttons. Diabodies or cis diabodies or mini-bodies. You can see you can take a parental antibody with pretty much any specificity that you're interested in, reformat it into a diabody or cis diabody or a larger mini-body fragment, radiolabel with your choice of different positron-emitting radionuclides. We even have fluorine-18 labeled diabodies and cis diabodies that work very well for same-day imaging and truly have either same-day or next-day imaging in these preclinical models. So that's the platform that we're using. And, but I also wanted to mention we're not just interested in radioactive imaging. There are many other ways you can combine the specificity offered by antibodies and employ these in different optical or multimodal imaging approaches. So I just wanted to highlight 3 of them here. The first one is a fun collaboration we did with Sam Gambier's group, where we did a combination PET and bioluminescent probe. We made a fusion protein of that anti-CEA diabody to an engineered stabilized rhodamine luciferase, and we're able to radiolabel it and show both by bioluminescence imaging and then also by PET imaging that protein did get to the CEA positive tumors. So that's combination PET and bioluminescence. More recently, we have tackled PET and fluorescent combinations. So here is work showing a dual-labeled PSCA mini-body fragment. And it was site-specifically conjugated to a fluorescent dye Cy5 and then radiolabeled with I-124 for PET. And you can see both in the PET imaging here, we have specific targeting to the PSCA positive xenograft, and then you can also see in this fluorescence imaging where the skin has actually been pulled back, the positive tumors have also clearly taken up in an antigen-specific way that fluorescent probe. And then finally on this slide I show a recent and really I think very powerful imaging application. This is the work we just had a minor contribution to this, but it's something I've wanted to do for a long time. This is work by Hung-Jie Dai and his collaborators up at Stanford, working in the near-infrared 2 window with fluorescent nanoparticles. And I had always wanted to put these cis diabodies onto little near-infrared nanoparticles and be able to do multicolor imaging. So he finally has done it. He uses structured light sheet microscopy, that the setup is shown here, and so he's able to image at the single cell level in tumors in living mice. And what he did was to label, I think in red is an anti-CD8 cis diabody coupled to a near-infrared nanoparticle, and in the green is OX40 specific coupled to a different nanoparticle. Both of these nanoparticles emit in the 1600 nanometer range, so they're in the near-infrared 2 window, but he's able to distinguish them due to the fact that they have different fluorescence lifetimes. And these are tumors that were treated with CpG, which is an immunostimulant, and I think the interesting conclusion they came away with is that you can see in the living tumor, in the living cell, on these immune cells, they are OX40 positive, but the OX40 actually was not on the CD8 cells. It was on the CD4 cells. So, but now we can tell this in living animals, non-invasively. Really very fun project to be part of. Okay, now I'm going to go way back because I've shown you here immunoPET, so that's applied immunology. But how are we going to apply this to immunology, which really has been, to me, the most exciting work that we've been doing in recent times? And I'm going way back to when I first started at City of Hope. One of my tasks was actually, I had a joint appointment in pathology. And this was so long ago that City of Hope was known as a center for the diagnosis and treatment of lymphoma. And that's largely due to Henry Rappaport, who actually is the person who developed the Rappaport classification of lymphomas, which was based on morphology. It was based on looking at H&E slides and what did the cells look like and how were they arranged, what patterns were they in. and they would classify lymphomas just on the basis of looking at those slides. And at that time, then the new development was immunohistochemistry. And so I was sitting down with the pathologists every week. And for example, if they had what they thought was a B-cell lymphoma, they would stain. So humans either have kappa or lambda light chains on their antibodies. But if you have a malignancy, it's monoclonal. It's going to be all kappa or all lambda. And so they would do immunohistochemistry, and here's an example showing that this was a monoclonal kappa light chain, and that's one way that they could diagnose the fact that this was malignancy because it was monoclonal. And my job was to bring molecular biology into this. So we were doing southern blots, looking at my favorite proteins, antibodies, and rearranged antibody genes, because again, seeing that rearrangement in these southern blots, I think you could see them, these little R's for the heavy chain, and in this case, the kappa light chain rearrangements tells you that you've got a clonal malignancy growing out. So this was a way to diagnose not only that it was a malignancy, but also a B-cell malignancy because of the antibody genes that are getting rearranged. So that was cool. Spent a lot of time with the pathologists, looking at a lot of slides, stained with a lot of cell surface markers for different lineages, of B cells, T cells, helper versus cytotoxic T cells, macrophages. So I became very familiar at that point, before I even got into the antibody engineering world, with the fact that in hematopoiesis, we now have very well-defined differentiation and lineage markers, cell surface markers with very well-defined antibodies that recognize them. So that just kind of stuck in the back of my mind for a long time. But then many years later, when I was at UCLA, Tony Rebus, who was a melanoma immunotherapist, he was one of the leading investigators, for example, in the development of the anti-CTLA-4 as a clinical treatment for melanoma. He led some of the large clinical trials that showed that it was effective. But early on, he came to me because this was at a time where we had FDG-PET. We could image tumors on the basis of their elevated glycolysis. But when you treat patients with immunotherapies, you also hopefully get an immune response. And those immune cells are also activated and glycolytic and will light up on an FDG-PET scan. And for example, one of the reasons you get a false positive in an FDG-PET scan might be infection or inflammation, as we know from some of the other work that's been going on that we've heard about this week. So he came to me and said, how can we distinguish these rapidly growing tumor cells from the activated immune cells? Because one's bad and one's good, and we need to know. And I said, I know how we can do that. We can look at the cell surface markers on the immune cells using immunoPET. So my lab, in collaboration with Tony and a number of other investigators at UCLA, did exactly that in mouse models. We made engineered antibody fragments that recognize CD8. So these are shown in a normal mouse. You see it's a mini-body, so you see clearance to the liver, but you see the spleen and a beautiful map of all the lymph nodes in the mouse. Here is our CD4 radiotracer. In this case, it was the smaller fragment, the diabody, so it clears via the kidney. But again, a beautiful map of the spleen and the lymph nodes in a normal mouse. Here also is a series of antibody fragments that recognize human CD20. So this is an example actually from Kirsten Zetlitz, who's sitting right over there, where she's imaging human CD20 in a transgenic mouse that expresses human CD20, and you can see the spleen and lymph nodes and bone marrow in this mouse. So we have these, we can make these radiotracers for these immune cell subsets. And so we decided, okay, where are we going to go with this? And, of course, Tony tells us, well, you really should be looking at the CD8 cytotoxic T cell, because in these immunotherapies, it's that CD8 cytotoxic T cell that is the actual drug. So to make a long story short, over the past several years, we've really focused on imaging CD8 cytotoxic T cells and other CD8 positive cells. And these are our workhorse agents at this point. On the mouse side, our lab is working with cis diabodies that are, as shown here, that are site-specifically conjugated and radiolabeled with zirconium-89. We have markers that are pan-specific across all strains. We also have markers that are strain-specific. Because it's a cis diabody, it shows rapid renal clearance, so you can see here the kidney activity. And then on right is the human-specific agent. which was developed by Imaginab, and it has a mouthful of a name, IAB22M2C, but now it has a different mouthful of name, has an official name, crefmurlimab. This is a humanized human CD8-specific mini-body with very high affinity, 0.4 nanomolar. So it should be non-immunogenic in people. It's biologically inert, as I explained earlier, and it shows rapid hepatic clearance. So here on the right is an example of imaging in a normal but humanized mouse. So what we see here is the fact that there is, we're able to detect the human CD8 T cells, or the CD8 positive cells that have taken up residence in the mouse spleen. So I'm going to summarize a lot of work by a lot of wonderful collaborators here, because at this point, this approach for imaging cancer immunotherapeutics first has been evaluated in a number of different approaches and systems. So I'll just show you the list here. We and our collaborators have used the CD8 imaging to evaluate immune responses in checkpoint inhibitor therapy models, in adoptive cell transfer models, combination therapies, oncolytic viral therapy, and also bispecific antibodies that are able to recruit T cells to tumors of interest. And also, because inflammation is key to many disease processes, we've also looked at additional non-oncologic applications. For example, stem cell reconstitution or graft versus host disease. So this kind of gives you an idea of how broadly useful it might be to be able to look at immune responsive, non-invasively on a whole body basis. So here's where we really want to go, which is clinical translation. And in this case, in addition to my academic research village, we actually needed an industry, a commercial village to help with this process. So these are reagents that were developed by Imaginab, but we also have to have wonderful clinical collaborators. So this is Nita Pandit Taskar, and I just wanted to show you just the highlights of some of the work that we've done with our academic colleagues. This is actually a PSMA mini-body labeled with zirconium-89, which was taken actually all the way into phase 2 in prostate cancer patients. But here's one of the, some phase 1 studies showing you the ability of not just those PSMA peptide analogs that Marty told you about on Wednesday opening session, but PSMA antibodies and engineered antibody fragments can also be very effective agents for lighting up PSMA-positive prostate cancer. For example, this one on the right is a patient where you can see the primary, but you can also see these very small lymph nodes. This was a pre-surgical patient that turned out to be definitely involved with disease. And then, of course, on the CD8 side, this is the CD8-specific humanized mini-body, and in the initial first 6 patients, this shows you a whole body scan of a patient. Now we're looking at the CD8 T cells, and you can see normal spleen, bone marrow, lymph nodes, and in this case, we see co-localization of that CD8 signal in a lesion, a melanoma tumor in this person who was on checkpoint inhibitor therapy. So at this point, the phase 1 study is complete and was published earlier this year. And what we can tell you about imaging CD8 in patients was that, as expected, we saw rapid clearance, primarily hepatobiliary. We did see uptake in where we expected it, in the lymphoid tissues, spleen, bone marrow, and lymph nodes. These are all 15 patients, and the thing that'll jump out at you first will be the spleen. No to low uptake in normal organs, muscle, heart, brain, and lungs, so we have low background in these normal tissues. And tumor uptake was seen in 2/3 of the patients, and it was variable, as you might expect. And the purposes, of course, of this phase 1 study were a dose escalation study from 0.2 milligrams of protein to 10 milligrams to see what would the optimal protein dose be. But then also these patients were imaged serially to try to identify what the optimal imaging time would be. And that could be as early as 1 to 2 hours after injection, although we've settled on 24 hours. So I wanted to show you some unpublished work as we continue to explore and validate, first of all, but then explore the potential utility of being able to image immune responses. So this is the phase 2 baseline and on-treatment study, where this is, in this study, patients who are scheduled to initiate standard of care checkpoint inhibitor therapy undergo a CD8 immunoPET scan prior to starting treatment and a biopsy at that time. They start checkpoint inhibitor therapy, and 4 to 5 weeks later, they have an early on-treatment CD8 immunoPET scan and a biopsy. And what I'd like to show you here are some examples from that study. First patient, a non-small cell lung cancer patient. The second one, a renal cell patient. And in both of these cases, I think you can see that pre-treatment and on-treatment, there was an increase in the CD8 signal in the tumors of these patients. Either this person was treated with a checkpoint inhibitor in combination with chemo, and then the renal cell patient, you can see nivolumab, just the checkpoint inhibitor pre-treatment and on-treatment, we see an increase in the CD8 PET signal. But importantly, if you look at the corresponding biopsies and the density of CD8 T cells in those biopsies, you see a concomitant increase in the CD8 signal. So the results of the correlation analysis of this study will be presented actually next month by Michael Postow at the Society for the Immunotherapy of Cancer meeting. So I think everybody's looking forward to, again, as Jason was talking about, validation that the signal that we're seeing in the PET scanner actually corresponds to reality. And finally, I want to show you one really cool slide from a really cool collaboration with Simon Cherry and Nagar Omidvari up at UC Davis, where they have that whole body U-Explorer PET scanner. And they did a study looking at patients following infection with SARS-CoV-2. And it's really fascinating because on the left you see a person who's recovered from COVID-19, and on the right a normal control. And I think what is striking is that in their patients they saw still, weeks or months later, very high levels of CD8 signal in the bone marrow and in the normal lymphoid tissues of these patients. So I don't know what this is telling us, but it's really interesting, thought-provoking. And the point I really want to make is that we're able to see things now, non-invasively, that we've not been able to see before. And it's going to be, raise new hypotheses and new questions. And really that's what it's all about. It's about continual exploration. So I'm going to wrap up here and say, I hope I've explained to you why antibodies and why radioisotopes and how to put the two together and how we can apply immunology to study immunology. But I really want to I want you to go away with the thought that as a group, we are inventing new tools and technologies that allow us to observe biology in action in ways that we've never seen before. So keep up the good work. I have so many people to thank. My early days at City of Hope, so here are some of my collaborators. My lab at City of Hope and our annual fishing trip, which is always great fun. And then also at UCLA, my two great mentors, Mike Phelps, Sam Gambier. Here is the group at the Crump Institute in 1998. And also, this is myself and Rob Ryder, who we've collaborated for many years on PSCA. And that is us sitting in front of a 2,000-page investigational new drug application for the radio-labeled anti-PSCA minibody. Nowadays, I hear sometimes you have to take a whole truck full of paper well, they don't use paper anymore, thank goodness. But I think the documentation is even worse, way worse now, to get something into the clinic. So that's something we really need to work on. I also want to thank my many collaborators. I'm sort of an honorary member at Stanford. I am actually an external member of the molecular imaging program at Stanford and have had many wonderful collaborations with Sam and his colleagues over the years that continue to this day. And then, of course, collaborations with many people in other institutions. I hope I've caught most of the main ones, but there's so many people that we've collaborated with. And truly, as a field, that's really how we advance, is by taking people with expertise in different areas and coming together and collaborating. And then, of course, for the ultimate clinical translation, I have to thank my co-founders at ImagineAb, past and present members of the team at ImagineAb, who are really doing a heroic effort in terms of taking these newfangled molecules all the way through into the clinic and hopefully to make them broadly available to clinicians and patients everywhere. And of course, we have to thank our patients and families who many of them in the middle of a pandemic volunteered for these imaging studies. They have no therapeutic benefit at all, but they're still out there and willing to participate in these imaging studies with the hopes of advancing our knowledge. And so many people to thank. And so all of this work was not done by me. It was done by the members of my laboratory. So I've listed the postdocs and graduate students on the slide. I hope I've got everyone, but there also were innumerable, it seems, undergrads, summer students, high school students, visiting faculty, all interested in finding ways to move this forward. So here is the lab, not just at lunches and lab meetings, but also our ski trips. And then at UCLA, we actually were a scary bunch. We were the group that was responsible for running the annual Halloween pub, and really it was a competition to see how crazy people could get each year. And I had to show this picture because it's from WMIC in 2015, and this is the whole family. We have Richard Tavare is the intact antibody, I'm the minibody. We have Amanda, who is the single chain FV, and then Kirsten and Wenting, who are the diabody, and then of course Kirsten's mom explaining that this is our lab. So that was a really fun WMIC back then. Okay, and last but not least, oops, I'm missing a slide. What did I do? We lost my last slide. My last slide has my family on it. It starts with my dad, who was an organic chemist, and he's the one that really inspired me to a life in science. And then there's this wonderful picture of my husband, Jeff, who flew all the way from Los Angeles to be here today. And also my children, my 2 daughters and my son, and now my son-in-law and our granddaughter Liliana. And my kids and my husband have put up with so much to enable me to be able to do this work, the long hours, the travel. I absolutely could not have done this without their support. So, huge thank you to you. Thank you to all for listening. I hope it was moderately entertaining. If nothing else, you kind of laugh at the antique way we used to publish papers and do research. The field's come a long way, but I think the future is very bright. We have much work to do. So thank you again.